So it's my great pleasure to invite our final speaker of our session today, Dr. Kei Kishimoto. Kei is the Chief Scientific Officer of Selecta BioSciences, a biotech company developing tolerogenic ImmTOR technology to mitigate unwanted immune responses, which is currently in phase 3 trials. Prior to joining Selecta, Dr. Kishimoto held leadership positions of responsibility at Momenta, Millennium, and Boehringer Ingelheim. He received his doctoral degree in immunology from Harvard and his postdoctoral training at Stanford. And the title of Kei's talk is Amplifying the Effect of Antigen-Specific Tolerogenic ImmTOR Nanoparticles with Engineered Treg-selective IL-2. So thank you very much indeed. We're looking forward to hearing your talk, Kei.
Great. Thank you for the introduction and the opportunity to present today to you guys. So I actually presented to this conference series. I look back, it was back in 2014, so it's been a while, but we've made a lot of progress on what we call our ImmTOR technology to induce antigen-specific immune tolerance. And as Andrew indicated, we're now in phase 3 clinical trials in a trial that reads out in Q1 of 2023. So what I want to do today is I'll briefly recap our original ImmTOR platform, but what I want to focus or introduce to you today is kind of our next generation of the platform, which we call ImmTOR-IL. So ImmTOR is a biodegradable nanoparticle that encapsulates the small molecule rapamycin. So rapamycin is an approved marketed drug for that inhibits the mTOR pathway, and it's proved for preventing graft rejection. But in the context of the nanoparticles, we're actually targeting antigen-specific immune tolerance. And the way this works is that we co-administer the nanoparticles with antigen to confer selective tolerance to that co-administered antigen. And we've shown that for a variety of different types of antigens, including autoantigens, therapeutic enzymes, as well as AAV viral vectors. And I don't have time to go through all of the background information, but we have been busy publishing a number of papers on the technology, and these papers can be found on our website if you're interested. So why do we use nanoparticles? The reason is really to leverage the natural biodistribution of nanoparticulates in the body and to deliver the particles to antigen-presenting cells in these organs. So specifically, when we inject these particles intravenously, in this case, it's a mouse, and it's a fluorescent labeled particle, you can see selective accumulation of the particle in the spleen and the liver. And within these organs, the particles here shown in red co-localize with these dark blue cells, which are the dendritic cells in the marginal zone of the spleen. They can also be found in macrophages, which makes sense because these are the endocytic cells, and they also happen to be the ones that present antigens to T cells. So the idea is that when dendritic cells take up the nanoparticles and see antigen, these the particles confer context to the antigen such that these dendritic cells become tolerogenic, and they present antigen in a way where we get formation of antigen-specific T-regs instead of effector T cells. You can kind of think of these particles as a tolerogenic adjuvant. So in a traditional vaccines either have exogenous adjuvant or they have endogenous adjuvant activity. And what that adjuvant activity does is it stimulates the dendritic cells to induce this more stimulatory response. Our ImmTOR nanoparticles, again, are acting like a tolerogenic stimulus that preferentially allows for this right-hand pathway to give us immune tolerance. We've shown this tolerogenic activity in multiple animal models. Here I'm showing data from the mouse where we're looking in the liver And in the liver, one of the main antigen-presenting populations is actually endothelial cell called the liver sinusoidal endothelial cell. These are among the most endocytic cells in the body, and they can present antigen to T cells. And when we treat animals with ImmTOR, these LSECs have gain a very tolerogenic phenotype, meaning that they've upregulated PD-L1 and downregulated co-stimulatory molecules CD80, CD86. When we take these LSECs from these treated mice and then co-culture them with T cells that are activated with CD3 and CD28, we can see that the ImmTOR treated LSECs can inhibit activation of T cells, as well as antigen-specific proliferation when we look at antigen-pulsed LSECs. Importantly, we can also show the induction of antigen-specific regulatory T cells in vivo. In this experiment, we're encapsulated an antigen called 2W. So we have nanoparticle encapsulated 2W, or nanoparticle encapsulated rapamycin, or nanoparticles that encapsulate both the 2W antigen plus rapamycin. So we give 3 weekly injections, and then 2 weeks later, we challenge the mice with the 2W antigen emulsified in complete Freund's adjuvant, a very potent adjuvant. And we look for the induction of endogenous T-regs that are specific for the 2W using a 2W specific MHC tetramer. And what we see is that only the animals that were treated with nanoparticles containing both the 2W antigen plus rapamycin induce these antigen-specific T-regs that are stable even with the challenge of antigen and complete Freund's adjuvant. We next looked at the specificity of this effect. So in this experiment, we have rapamycin plus the antigen ovalbumin compared to a particle that just has rapamycin, in which we don't administer any co-antigen. So we give an injection here on day -14, and then the mice are immunized with both ovalbumin and KLH 3 times. And then we bleed the animals on day 41 and look for immune responses to both ovalbumin, which is the dark teal bars, and KLH, which are the orange bars. And if we look at the group here on the far right, the groups treated with both nanoparticles containing both ovalbumin and rapamycin show inhibition of the anti-ova response without affecting the antibody response to KLH. And importantly, if we just give the rapamycin particle without antigen, there's no effect on either the anti-ova response or the anti-KLH response, indicating that the nanoparticle is not just acting as a slow release form of rapamycin that's causing general immune suppression. You have to give the antigen at the same time that you give the nanoparticle. So in the clinic, we've been studying ImmTOR in combination with a very immunogenic enzyme therapy called Pegadricase. This is a fungal-derived uricase enzyme that is used clinically to metabolize uric acid for the treatment of chronic refractory gout. So this is a tetrameric fungal enzyme. And as you can imagine, most patients develop antibodies very rapidly to this enzyme therapy. And I'm showing you data from our initial phase 1 clinical trial. And here, given a single dose of the enzyme by itself, all of the subjects develop antibodies within 2 weeks after a single dose. And when we look at serum uric acid levels, so these patients were selected for serum uric acid levels around 6 at baseline, And we see after treatment a very rapid reduction in serum uric acid levels that stay low for 7 days, but by 14 days, it starts to rebound. And by 30 days after that single dose, 4 of the 5 subjects are back to baseline levels. In contrast, when we add increasing dose levels of ImmTOR, we see a very nice dose-dependent inhibition of that antibody response to this enzyme. And when we look at serum uric acid levels, we see a correlating reduction, sustained reduction in serum uric acid levels over that 30-day period. And so we went on into phase 2 clinical trials and showed that by giving multiple monthly doses of this combination, you could have sustained reduction in serum uric acid levels in the majority of patients. And as I mentioned, we're in a phase 3 program, which consists of 2 different studies. This study has been fully enrolled. The last patient visits will be towards the end of this year, and we'll have top-line data to announce in Q1 of 2023. So I want to now focus on kind of the next evolution or generation of the ImmTOR platform, which we call ImmTOR-IL. And this is the combination of ImmTOR plus a engineered Treg-selective IL-2 molecule. So interleukin-2 is an important cytokine for both effector T cells and regulatory T cells. However, these cells use different receptors for these effects. So effector T cells express an intermediate affinity form of the receptor, which consists of a beta and gamma chain, whereas regulatory T cells express a trimeric receptor complex that's comprised of alpha, beta, and gamma chains, and this is a high affinity receptor. And so various groups have been able to engineer IL-2 that are either selective for T-regs or T effector cells, which are being developed for cancer therapy, or for selective for the high affinity form on regulatory T cells, which are being developed for autoimmune diseases. And of course, we've been more interested in these T-reg selective IL-2 molecules. So the way that these IL-2 molecules work is that being selective for T-regs, they can expand total pre-existing T-regs. So in other words, these teal color cells represent T-regs. When we treat them with an engineered T-reg selective IL-2 molecule, we see expansion of T-regs. And the idea that companies are pursuing is that by expanding total T-regs, that you might be able to increase bystander suppression that would be beneficial in autoimmune disease. Of course, our approach has been more about antigen-specific T-regs using ImmTOR plus antigen, where we can induce T-regs of novel specificity to, for example, to this fungal enzyme. And this is depicted here by these kind of dark teal color cells. And the question we had was, if we combined ImmTOR and antigen with an engineered IL-2 molecule, could we see selective expansion of antigen-specific Tregs? And so we tested this in a mouse model where we adoptively transferred ovalbumin-specific OT2 cells and then treated these mice with ImmTOR plus ovalbumin or IL-2 mutein plus ovalbumin for the combination. And we first looked at total Tregs in the spleen. So if we focus first on these turquoise colored bar here in the middle, this is the ImmTOR plus ovalbumin. And we don't see expansion of total Tregs, which we knew coming into this. However, in the next bar over in orange, when we treat with an IL-2 mutein plus the ovalbumin, we see an expansion of total Tregs, which is consistent with what's been described in the literature. However, when we combine the two components, whether or not there's ovalbumin, we see a further bump in total T-regs, which we attribute to possibly due to increased survival of these expanded T-regs. Next, we looked at antigen-specific T-regs. So again, starting with the turquoise colored bar in the middle, when we look at ImmTOR plus ovalbumin, indeed we see an induction and increase in ovalbumin-specific T-regs, which is exactly what we would have expected. And we know this magnitude of expansion is clinically meaningful based on our gout program. However, when you look at the IL-2 mutein plus ovalbumin, there's really very modest, if any, expansion of these antigen-specific T-regs. But when you combine the three components of ImmTOR plus IL-2 mutein plus ovalbumin, we see this massive expansion of antigen-specific T-regs. And we think that this could be very meaningful in settings such as autoimmune disease, where you have large numbers of pre-existing antigen-specific effector T cells, and you're trying to offset them with reduction of antigen-specific T-regs. So we tested this combination in a mouse model of gene therapy. So here we're using AAV viral vectors, which not surprisingly are immunogenic. And we're looking at the immune response to the AAV vector. So in this experiment, we give 2 doses of the AAV, a first dose on day 0 and a second dose on day 56, plus or minus the ImmTOR and plus or minus the IL-2 mutein. This group here is the group that's just treated with the 2 doses of the AAV. And what we're looking at is the anti-AAV antibody response over days. And as we would expect, you get a very rapid and robust antibody response to the AAV viral capsid. Now, if we add the IL-2 mutein to this mix, so again, 2 doses of the IL-2 mutein on day 0 and one on day 0, one on day 56, we see some attenuation early on, but it's very modest and it's not durable. When we add ImmTOR, so in the mouse, the therapeutic dose of ImmTOR is 200 micrograms. We see very nice inhibition of antibody responses to the AAV. And even after the second dose, we see inhibition, but we do see breakthrough in antibodies in some of these animals. And if we use a suboptimal dose of ImmTOR, we see this breakthrough happening earlier in the response. What was really exciting for us is when we combine, again, the ImmTOR with the IL-2 mutein, again, just 2 doses, day 0, day 56, we see complete inhibition of the antibody response through the end of the experiment at day 117. And this happened even at these suboptimal doses of ImmTOR. So even at this 50 microgram dose of ImmTOR combining with this suboptimal dose of IL-2 mutein, we see complete inhibition of that response. Next, we looked at a higher vector dose. So in this previous study, we were looking at doses up to 5e12 vg per kg. So here we're looking at a dose of that's 5 times or 10 times higher, 5 times 10 to the 13th vg per kg. And not surprisingly, this is a huge antigenic challenge to these mice, and results in a very strong immune response. When we give a single dose of ImmTOR on day 0, we see pretty good inhibition out to the first month, but then we do see these breakthrough responses happening in the majority of the mice. If we give monthly doses of the IL-2 mutein, so 4 monthly doses, we see, again, some attenuation, but it's pretty modest and incomplete. But again, when we do look at the combination of a single dose of ImmTOR plus monthly doses of the IL-2 mutein, we see very nice inhibition of the antibody responses out to day 131. So we're excited by this combination, not just for the increased efficacy, but also for potentially this could be one of those rare cases where the combination is actually safer than the individual components. And I say this because the data I just showed you shows that by adding the IL-2 to ImmTOR, we can have dose sparing of the ImmTOR component. And then for these IL-2 muteins, the biggest concern with the IL-2 muteins in the clinic is that at higher doses, you start getting activation of effector T cells. But we know that rapamycin inhibits proliferation of effector T cells, but not T-regs. So this could increase the therapeutic window for these IL-2 muteins. And finally, we know these mutated molecules have the potential to be immunogenic, and we know that ImmTOR can mitigate immunogenicity of co-administered biologics. And finally, the synergistic expansion of T-regs, again, could be very important for its activity, both for use in mitigating immune responses to biologic therapies, as well as for treatment of autoimmune disease. So in summary, we have our original ImmTOR platform, which has shown very robust activity in our gout program. And again, we have a phase 3 program that's reading out at the end of this year. We're working on our own proprietary IL-2 molecule in partnership with Cyrus Biotech, which is a spin-out out of David Baker's lab, which is one of the premier immune or protein engineering companies in the world. And then we're really excited by this combination, which has can not only expand total T-regs through the IL-2 component, but can induce and expand antigen-specific immune tolerance. So with that, I'd like to thank the team at Selecta, and happy to take any questions if there are any.
Very nice. Thank you indeed, Kei. It's great to hear your presentation. Thanks for sharing that with us. We have a question here from Steve. I don't know, Steve, you want to ask it verbally? You can unmute.
Yeah, I was just wondering why you think you have to dose monthly with the ImmTOR for Pegadricase. Do you think it's due to the half-life of the inducible T-regs?
Well, there's a couple reasons. I think one, we're giving we want to give multiple doses of the ImmTOR with the antigen to kind of reinforce the memory of the tolerance, so that it's not so dissimilar to giving booster shots for a vaccine. But the other is just to kind of maximize the effect over time. So in gout, this isn't meant to be a chronic lifetime therapy. There's a lot of oral therapies available for gout, but what the uricase allows for is to remove these very large deposits of urate crystals that accumulate in joints and tissues, and that's the cause of pain in gout. So if we can debulk these patients of these urate crystals using a course of therapy of probably about 4 to 6 months, then these patients should be able to go back onto oral therapy.
Thank you. Quick question. In the context of the IL-2 that you're talking about, I mean, do you envisage, or just adds to the complexity, but do you envisage the possibility of adding any additional cytokines to get increased benefit?
I mean, at this point, we're not thinking of adding anything else to it. For one, as you say, it would be increasingly complex, but two, you know, we'd have to really be built on a compelling scientific reason to combine. And right now, we do see this really strong synergy between the IL-2 component and ImmTOR, and that's what we're building off of. So it's not just an additive effect; it's truly a synergistic effect.
Yeah, no, that's great. Very nice. Brilliant. Do we have any other questions at all?
I would like to jump in. That's okay.
Go for it, Liv.
So, it's all of the T-regulator cells that are being upregulated specific to that antigen, or is there any impacts on other non-antigen specific T-regs within that system?
Yeah, so that's a good question. So I showed you with ImmTOR by itself, we're only affecting antigen-specific T-regs. But when we add the IL-2 component, we are seeing increases in total T-regs in addition to antigen-specific T-regs. But for treating like autoimmune disease, we think that could actually be beneficial. As I mentioned, there's a number of companies, you know, a lot of big pharma companies like BMS and Amgen, Lilly, that are developing these engineered IL-2 molecules with the idea that they're only expanding total T-regs, but there's some compelling data that these could be beneficial in autoimmune disease.
Thank you.
Okay, any further questions at all? Any raised hands? I see none. Great. Well, thank you very much indeed, Kei. Really appreciate again your talk. Thank you for joining us.
Thank you.